Alright, in this video I'm going to go over essentially how to do your entire annotation for the lac operon. You've done an annotation already once, and we took our time and did that in such a way that I think you've got a pretty good idea of what you need to do. Here are the steps needed to do our lac operon, and specifically a bacterial gene. So the first thing we need to talk about is that there is something called a Shine-Dalgarno sequence. And this is the place where essentially the ribosome sticks to the RNA. Now, this is actually one of the easiest things to find. Realize that they're kind of like they're a signal for the ribosome to bind to the RNA. So of course that means if they're in the RNA, then they're also, the complement will be in the DNA, and you can find them. So they're actually just a bunch of A's and G's. Now of course, where is this? Is this in the RNA? We don't know. There are no U's or T's here. So is this in the RNA or the DNA? All right, so this is where the consensus sequence where it binds to the RNA. So if you're looking for this in a DNA sequence, you would be looking for T's and C's, but if you're in the RNA, you're going to be looking for A's and G's. And it does not have to be in this sequence. This is just the consensus. This is the most often found, all right? But it usually has an AGGA in it, but it doesn't have to. A GAGG will work just as well. In other words, it's just a series of A's and G's. So if you find a bunch of A's and G's together in, say, as few as 4, but looking for like 6 or more, you're probably at a Shine-Dalgarno sequence. So a Shine-Dalgarno sequence, really, to be honest, is just a bunch of A's and G's near each other. If you see 6 or 8 A's and G's together, you can probably call that a Shine-Dalgarno. Now, you don't know unless you go in the lab and see if a ribosome will stick to that piece of RNA. So the difference here, of course, is, remember, in a bacteria, the ribosome sticks to the RNA. It isn't like the RNA goes out and finds a ribosome. Now, in a eukaryote, in our genes, we make RNAs and they go out and they have the 5' cap. That 5' end will stick to the ribosome. Here we look at it differently, and we say that the ribosome goes to the RNA and actually looks for something like this sequence. And you can get multiple ribosomes attaching to the RNA at the same time. So you can be translating, all right, the RNA by multiple ribosomes along the RNA at one time. Completely different. This is the thing that is probably the most confusing. People getting this kind of idea that you can have an RNA with multiple ribosomes attached to it. This is not how it works in eukaryotes, okay? Now, where do you look for these things? You look for them from 3 to 13 base pair upstream from a start codon. So if you're pretty sure you found your start codon, look anywhere from 3 to 13 upstream and look for something that looks like this. But all it has to do is be a bunch of A's and G's, all right? No C's and T's. And this is going to be in the RNA. You could look for it in the DNA as you're doing it. You don't have to translate to the RNA. Just remember that you would be then looking for T's and C's. And here's the location. So we know this very well. We know it. I think I'm going to show you one where actually this runs into an AUG. In other words, I think the A from a start codon is actually part of the Shine-Dalgarno sequence. I think I will show you an example of that in a future video that I've made several years ago that I'll have you watch. But it means it can really be like this 3. It can be right up against the start codon. It doesn't have to be 3. All right. So all of these things you should keep in mind as you're doing this. Whenever I tell you it's this, that is just I'm going to call it a guesstimate. All right. And it's why you can't have a computer do it. You can't say look for it with right here, computer will look for it here and it won't be there. And look for just a bunch of A's and G's. Most people probably put in, oh, look for this. Well, you're not going to find that most of the time. You're just going to find a bunch of A's and G's near each other. And so it does take some human judgment. Now, the Shindell-Garneau and the start codon will be within a 30 base pair conserved region due to the structural limitations of the ribosome. Now, this one is true. This one you can go by. There's about 30 base pair, all right, and you pretty much have to have the start codon within 30 base pair of the Shindell-Garneau, or the ribosome won't work, all right. This is kind of a set in stone thing of the way we know. So here's something that is a bit more structured, is this 30 base pair conserved region. And within that 30 base pair, you're going to find this, and you're going to find the start codon, all right. That's just, so this is actually a pretty easy one to find. If you kind of know the general idea of where, you don't even have to know where your start codon is exactly. You just have to know kind of the region where the gene starts and start looking for a bunch of A's and G's, and then look to see if within, 20 base pair up front here, or, go 30, look to see if you find an AUG. And if you do, you found it. Of course, now I'm about to tell you that AUG isn't always AUG, the start codon. So here's the problem. In bacteria and eukaryotes, I mean, you read a book and it tells you the start codon is AUG. That is not always the case, and in fact, I'm going to do an example with you in a video that I made a while back for this class, but where I show you that it isn't always AUG. So the second thing you find, look for this first in the right area, then look for your start codon within this area. So find this AG rich region in the right area, then look for your what? Well, you don't look for an ATG or an AUG, all right, in your FASTA file. You look for a TG. A start codon, even though this is methionine, and the thing is, even though these don't code for methionine, they'll end up being methionine if it's at the start codon. So a start codon actually just needs a TG. It does, the first letter is actually an N for a start codon, but they don't tell you that in intro books or even in graduate books. You have to just learn this from experience. All right, so keep this in mind, that a start codon can be any of these four. All right, so it can be an AUG, a UUG, a CUG. Now I did it with T's because, remember, FASTAs do not have U's in them. So you look in your RNA, all right, and in your FASTA, you're going to have T's. So basically, you have to go in and substitute U's here for these T's when you're in your RNA. So keep that in mind. All right. Then our reading frame. Well, the reading frame I think you've already gotten, and I didn't say much here in this review. Just realize your stop codons. You can find those as long as that. Now, luckily, the great thing about the bacteria, no splicing. We don't have to worry about it. So once you've designated your reading frame with your start codon, you can just go and read them 3-3-3-3. We don't have to worry about splicing here. Makes it so much easier. All right. Now here I go into a lot of details on terminators. And if I hadn't used the word, there is attenuators. And often a hairpin loop. In bacteria, we have it down a little better than eukaryotes. We were doing eukaryotes, and to tell you the truth, what you found in the GPX genes may or may not have been they've found it because they've done wet lab experiments, but it doesn't have, there are really no rules for terminators in eukaryotes, to tell you the truth. Here there are. I mean, you can find them, and you guys set out to find them, and I suspect a couple of you actually did find the terminator in our project 1. Finding the terminator in a prokaryote is going to be a bit easier I didn't say real easy, but a bit easier, because there are just a couple of ways to do a terminator. It's row dependent and row independent, and you can follow these rules pretty easily. Um, it's in eukaryotes, it's going to be a GC rich followed by about 6 U's. So, like I said, in eukaryotes, it can be T's, it can be which would be used. It really, you should think of the terminator as being part of the RNA, really. But you can, of course, just use the complement and use A's and T's. But here, you're going to look for a GC rich one, with followed by about 6 U's. So keep that in mind. This would, of course, translate on DNA as A's. If you're looking at it from the DNA side instead of the RNA side. But keep this in mind. This is how you find it. Much easier to find a terminator here. Like here they even tell you about the tripoperon, that you should look at that, and I gave you a tripoperon example in the for the quiz. Now, R dependent, are C rich, G poor. All right, and this is just a place where a protein, instead of the hairpin loop causing it to fall off, you're essentially going to have a protein that sticks there and causes it to fall off. So it's a different one. And notice it says it's at the are at the 3' end of eubacterial, so slightly different, not quite. For our E. coli, we should look for this one. All right, and that's why I looked at it. There is also an R factor. It uses ATP. This is for genes that you have to regulate a little bit more intensely than just a regular terminator. Not important for us, but know that it might be necessary, it may not. And just read it. We know about it now. It's not important for us because it's not sequence based. It just binds to free RNA. And if there's nothing going on there, it's going to stick to it, and it might cause termination. There really isn't much to it, except that it uses energy. So for a bacteria, that's a big deal. And it's for those regions that you have more, how do I say, dangerous proteins, that you might use this. When I say dangerous, you want to make sure you don't make too many of them, or it doesn't go on to be a longer protein than it should if somehow something interrupts the stop codon. All right, now moving on. I don't want to get bogged down in this because it's not important. I want you to know it, but you don't. It's not going to be important for our project. All right, the promoter region is very important. So this is the first time we really looked highly into the promoter. It is approximately, its technical name of a Tata box is the Prinbrow box, but nobody calls it the Prinbrow box. They call it the Tata box for obvious reason. There is also something called the Cat box, that's at minus 35, and it also often has a CAT. Often there is a T. It's TTGACA, and there's a T here. And so this is called the Cat box. And the Cat box and the Tata box work together. The Tata box is, actually you'll see books say it's at minus 10, others say minus 11. This one is usually at minus 35, and usually these are both there. And these regions are necessary for the termination of the, for the initiation of transcription. All right, so you have to have usually both of these in a bacteria. This is the primary one. This is the one that we're calling the promoter sequence. But really the promoter sequence is usually two sequences. It's a Tata box and a Cat box. And usually they have to be within a certain distance of each other. So keep that in mind, that you need to find that these two exist, and they're important. You need to find the cat box may not be there, and realize these numbers do not have to be right on. I think I'm going to show you an example where the the Tata box is more, if it's the one I'm remembering, the Tata box is more like a -6 or something. It's much closer, and but this one needs to be about 25 from it. So if that one's at -5 or 6, then this one is going to be what, at -30. They do tend to need to be a certain distance apart, but there is a 30 base pair region. If you remember up with this one, this 30 conserved here. There's a similar kind of thing going on here, that there's a kind of region that they have to be within a certain distance of each other. I've certainly never seen one that's more than, I've never seen one of these that are further out than -51 or 52. I've seen this one right on at 0, or at essentially -1. I've seen this one where it's like part of the start codon. So this could be anywhere from -1 to, I don't know, -15 or 20, I guess. And this one, the range could be really close. It could be up near, much closer at even something like 15, but it could also be all the way out to -50, and that just comes from me doing a lot of these. So there's a range in there, all right? Usually look at about 50, no more than 50 base pair before the transcription start site. Now, you guys don't know how to find the transcription start site yet, but generally transcription start is at +1. So this is at -10, so this would be 10 before the transcription start site. This would be at -35, the transcription start site. And 17 is also a rough estimate. All right, so don't set these in stone. And that's why you can't have a computer do it easily, because there's always the potential that it could be further away than whatever parameter you set. All right, there are some other features that you might find, like an up element. So look through these and see if you can find these. So when you're annotating, you need to look for an up. Just some more things that help regulate the gene. We're going to find this very common in eukaryotes. They're going to be things called transcription factors that will bind and regulate the gene. We don't see that as much in bacteria. They don't have the same kind of regulation that we have. They have more regulation within the gene that we're seeing, like the weak and strong terminators and things like that. Those are more of how a bacteria regulates its genes. We use transcription factors, all right? And the up element is similar to a transcription factor, so look for it. Here are your letters that you can look for it, and your locations. So this is just an extra sequence. Let's see, the sigma 70, you may have learned about that in molecular. But basically what we're looking for is something like this. So here is your gene to be transcribed. Here would be your -10 and your -5. What we're going through here is we're showing that the percentages of each of these letters, now they're not aligned, should have done a better job in making this, but your actual Tata box, 77%, if we're going to go 1, 2, 3, 4, 5, 6, if it's 6 letters long, 77% of the time, the first letter in your Tata box is going to be a T. Doesn't have to be. An A is there 76% of the time. Another T then is 60%. This one is 61. This one is 56. And this one is 82. So the strongest one is actually the last one. So there's a T here, the two strong ones. So there's a T here and a TA. So if I were looking at this, I would say we're looking for two T's that are what? That have 4 of something in between them. So if you're really looking for a Tata box, the strongest percentages here show you that it's a probably a TA. I would look for a TA, then skip 3 and 4 down, then I would look for another T. And so I would look for a TA, T, but these are all going to have to be TAs. That is pretty clear. Not always, but I would say very much so. But you could put anything in here. But if you find a TA, 3 of something else, and another T, you might put it as a potential TATA box if it's at the right spot. Now, the right spot, though, it isn't always -10, so it makes it just really hard to figure out where the TATA box is sometimes. But it's doable, all right? More often than not, you can, in a bacteria, you can find the TATA box. The -35, a lot fuzzier. Look at this. This is 77 and 76% and 82. You don't get anything near that good. There's a 79 here that this is a T. That is about it. And there's often a T right here. There's usually it's a CA and a T, and that's why it's called the cat box. But that in this, it's not high percentage. It's just that people have noticed that it's a CAT. So you got TATA and cat, and so it's been called the cat box, and they still call it that. So this is called the cat box, and it's kind of funny. So the TATA box and the cat box. And I've already gone through that you can't trust these locations, and you can't trust these percentages. They're just percentages. They're not set in stone. So this is how you draw or write out what you think the consensus is. So as a general rule, you can look for between -36 and -31. You can find a cat box, and it's going to look like this. Right here. All right, you might even add that extra T in here, but it can really be anything. But if it's a T, it's a C, and if there's an A here with a C here, and then you get a T here, it's cat, and so that's it. But the TATA box, all right, its consensus, the best one is TA. Like I said, a pretty high percentage of this A here, but these two can change. You pretty much need to know that this little T and this little A could just as easy be this A could be a T, and this T could be an A. So you don't have to do it this way. But this is the most likely one found. It doesn't mean that any of them even have this one. It's just that how you create a consensus where you find the most likely one across these. And they've found one that is the one that binds the best. But you find the one that always attaches, and I've forgotten it now. I think it's T. I've forgotten. But there's one that they artificially created, and it's a very rare one because when RNA polymerase attaches to it, it just always does transcription. It's very strong, and it actually isn't the most efficient one. So the actual one that the enzyme will bind to the best is not the one that is actually the most efficient at starting transcription. So that's always kind of interesting. It means there's probably still a few things we need to learn about how this works. Going down here to B, I'm not sure. Oh, this is a different promoter. This is not a TATA box. Realize that TATA is only one of many promoters. Now there are only a few in bacteria. This would be another one. Yeah, there's a C in here, so you don't count that as a TATA box. Probably slightly different proteins. Maybe a different conformation of a protein or something due to temperature, salinity. Might still be RNA polymerase, but maybe or maybe it's one of the other polymerases and it doesn't use a TATA. But this is the RNA polymerase 2 that we're essentially, or the equivalent in bacteria that creates mRNAs. That's what we find in bacteria. And here you might find something else. This is a slightly different version. But you can look for any of these, but I can tell you that you want to be looking for these in our genes. Now, sequences to look. These I'm showing you all of the potential promoters here. So, and the type. So as a general rule, TATA box is the most common. But if you're looking at a heat shock protein, you're going to look for something like this. If you're looking for the flagella genes that have to do with the promotion of the flagella, it has a promoter. Instead of a TATA box, it has this thing. This looks a lot like it could be a GC box. Maybe this is what evolved into a GC box. And here even, maybe these evolved. This one is for nitrogen starvation, a gene that has to do with when this genes that are produced whenever the bacteria is under nitrogen starvation conditions. Those genes, this is the promoter, the place where RNA polymerase will stick. All right, now here are some just general rules that I'd like you to go through and understand. There are some terms like cis and trans that you ought to understand. So make for sure you know what it means when I use the term cis. All right, and what I mean by the word trans. So a cis element, okay, I love this. Cis elements there are trans, in contrast, there are other molecular act on the molecule, for instance, a protein binding. Okay, how do you think about this? All right, cis is the sequence that's on the DNA. In other words, when something is actually on the thing that it's controlling, then we call it cis. It's on the same sequence. So a TATA, a TATA box, is cis to your gene. The RNA polymerase is trans. These are molecules that act on the molecule. So you think of the thing that attaches to the TATA box, which is RNA polymerase, that is a trans element. It is a protein that has to come in and do something to the DNA. Cis, all right, is something on the DNA that needs to be there for the trans element to act on. So your TATA box is cis to your gene. Your RNA polymerase is trans to your gene. So you need to get this kind of thinking in your head on how this works, because you use cis and trans. So I would say RNA polymerase is trans to the lac-operon. All right, but I would say that the TATA box is cis to your lac-operon. All right, the O sequence is cis. The I protein would be trans. All right, things like that. And you need to understand that terminology. All right, in prokaryotes, the promoter consists of two. Well, we've gone over that, so you might read over that, and it's called the Prembrow box. So keep that in mind. No one uses that term, but it's out there. It's TATA box now. This other one is, I should put in here, I should add someday that this is called the cat box, and there is often a T here at the end, just enough for them to call it the cat box, and I think they just wanted to call it the cat box because it was funny, but there should be a capital T here. The promoter selectivity is determined by the sigma subunit. You might know this from molecular. We're not interested in knowing that level of detail about the proteins. Leave that to molecular. But this might help you in molecular. All right. Let's see. Anything else? Yeah, just read through this. You don't have to know about genomic select screening. You can read it. Now this represents transcription factor dependent inducible promoters. You need to know this word constitutive, which means essentially always on. And inducible means it's off but can be turned on. This is like the lac-operon. It's usually off but can be made to be turned on. This one is turned on all the time and has to be made to go off. So constitutive versus inducible, you should know those. They're going to be on a quiz, so you should probably know the difference between those two. Let's see. Otherwise, kind of review this. This is what you will have to do for project too, with some other things. But this is how to get the basic annotations, and this is probably the order you should do it. I would look for this first in conjunction with your start codons, realizing that they don't. And I'm going to have a video where I go through this. I'm going to do the I protein for you. Then you find your reading frame. Then you find your terminator. All right. And then you look for your promoter. All right. And that would be the initial starting steps for annotating a basic bacterial gene. And I will do the I. Now, of course, if you do the rest of the operon, it's going to be considerably more dependent because you've got to find O, you have to have the cap, you've got weak and strong terminators, you've got all these things additionally find in there. And we have not talked about the all-important transcription start site, which will be another video next week.